All right. So we've had an exciting morning of several presentations, three, showing either single particle cryo-EM or X-ray crystallography, structural information and molecular dynamics on individual protein structure. And I want to just give kind of an overall vision for EMSL visual proteomics and take a few minutes for that. So Douglas presented the roadmap overview of the future for where we want to go at EMSL over the next 5 to 10 years. And the digital phenome is one of the major roadmap strategic directions that we're going. And of that, developing a visual proteomics platform for EMSL users is one of the key thrust areas of that, and has kind of a dedicated time frame of the next 3 to 5 years. And what we really describe as visualizing the proteome really comes from this driving force of, in order to be able to do proper functional annotation and phenotyping, we really also want to know the enzyme localization and quantity. And uncertainty of localization and quantity in single cells is really a bottleneck for getting clear genotype to phenotype interactions and other functional annotations. And so we really want to go between these two descriptive images. On the left-hand side, you have traditional models of compartmentalization and different metabolic pathways occurring in each one, versus on the far right, a cell pack model of packing all the known proteins into a cell of a given size and trying to understand the overall packing, interactions, and so forth. Basically, for the EMSL user program, the development of visualizing the proteome, or visual proteomics, as we're calling it, is really about trying to provide direct experimental evidence to support each one of these, and also allow us to drive towards creating a simulated virtual cell. Now, those are lofty goals, but one of the main reasons that we think that we are one of the facilities that are capable of pushing this forward, there are a few around the world, but for us, we wind up having top-down and native mass spectrometry capabilities to look at heterogeneity, stoichiometry, molecular interactions, post-translational modifications. We have SC proteomics, single cell proteomics to do spatially and cell resolved proteomics. We have cryo-EM for high resolution structural elucidation, as well as electron tomography to be able to get the whole cell context, and NMR solution phase structure determination, as well as having advanced computing for data integration and prediction. So all of these capabilities are housed within one building, as Douglas mentioned. And so because of these, this is really driving the framework for us to be able to push forward in visual proteomics. And as you'll notice, the creation of this integration meeting, the 5 different sessions that we have, is really about areas that link directly to each of these capabilities, as well as the overall goals. So being able to push the frontier of structural approaches to peer inside of cells and understand their ultrastructure, looking into the cell, the proteomics and the dynamics that are occurring in proteomics, post-translational modifications, and then using advanced computing to push those models, as well as visualization of data for more efficient interpretation. So that's kind of been the framework that we've been operating this integration meeting under. So I hope that you all will find some excitement in that as well as we go through the various days. But in order to take it the next level of, if I'm a user, what's available for me to access now? Since we're talking about a vision for visual proteomics coming online over the next 3 to 5 years, what can I really utilize right now in order to push my science forward? Well, as Douglas alluded to, EMSL has over 150 instruments co-located in a single building. You'll see that we have everything from super resolution fluorescence to optical microscopy to cryo-electron microscopy, even X-ray, microtomography, and nanotomography. We wind up having NMR, mass spec, EPR, cell isolation systems, instrument development lab, a whole bunch of capabilities in addition to microfabrication and microfluidics. The one thing that we really don't have is X-ray structural work. And so we have a large array of capabilities spanning a lot of different integrated research platforms that Douglas referred to earlier. And these are really the foundation of all of the user projects that are coming in. The molecular bioimaging one is one that we can really focus on for the rest of this talk. And if we just look at a sampling of some of the capabilities that lie in that, you can see that we have a large array of electron microscopes, as well as top-down proteomics, helium ion microscope. And the two on the far bottom right, the Krios G3i and the Aquilos 2, these are two new systems that we've recently installed and have been added to this overall integrated research platform and are available to users. I'll talk about both of these in just a minute. Now with the Titan Krios G3i, as I mentioned, we wound up recently installing it back in July. It's been fully commissioned as of end of September. For single particle imaging of soluble or membrane protein complexes, we've gotten a couple reconstructions at 2.2 angstroms and one so far below 1.9 angstrom resolution. Single molecule or small molecule microelectron diffraction and protein-based microelectron diffraction. The small molecule micro-ED we're able to do de novo structure determination with, as we're oversampling and wound up having diffraction spots out to about 0.7 angstrom resolution for that. And then whole cell tomography is the current workflow that we're fully commissioning right now. But you can see that we do have capabilities already in-house for this. So the Krios is already available to users. We've started a few user projects as part of the commissioning phase. We will be opening up a special call for BSSD funded and BER funded researchers who can apply directly. The other mechanism is to go through the normal EMSL user program proposal mechanism if you're going to be requesting access to any other instruments as well. But if it's just for the Krios, we have a rapid proposal mechanism available. And please reach out to me if you have any questions. Besides the actual cryo-EM instruments that we have and other direct imaging, we've also been developing approaches to overcome bottlenecks in linking proteomics workflows with structural biology. And this was really driven by a need that arose where we noticed that we would get many user samples, do proteomics on them, and in many cases, people would be very happy to get the protein abundance profiles. But in some cases, the proteins of high interest would be ones that have no known function or limited functional or structural information on it. And the question was then, how do we push that into the structural workflows? We used to have a gap with this. Besides doing E. coli expression, we wanted a faster route to be able to do that. To create that linkage. So we can go from creating a protein abundance profile, identify a target, and then rapidly send that to structural approaches. So we built a cell-free pipeline. This is showing you the general workflow for the wheat germ cell-free pipeline that we have. We've also in the past used both a human cell-free pipeline as well as a E. coli one. Both of them are available, but we highly prefer the wheat germ one. You can take any target gene, subclone it into one of our vectors, and then we wind up doing various scale expression. And so we have a mini, midi, maxi, and mega scale reactions. The mega operate in a fully automated robot that allows us to do cell-free expression. The mini expression scale allows us to do many different expression tests all at once on size scales of about 25 microliters. So we can take the target protein or target gene, put it into the cell-free system in the test tube, and with 25 microliters, screen various conditions to look at whether or not additional additives assist with the solubility or overall expression of the protein. And so we can do large scale screens of various components before we start to scale up in overall production. In this case, you can see on the upper right corner a gel showing an easy one-step flag tag purification from the expression lysate. Bottom right shows the ability to do functional assays with this. And then based on the various scales, the maxi versus mega, you can send for either cryo-EM or X-ray crystallization. And in both cases, we're able to get atomic resolution structure from it. And as you can see from the bottom left side, we've expressed proteins from bacteria, algae, yeast, plants, and humans with these approaches. And this cell-free pipeline is available to users already as well. I think we had 5 different user projects last year taking advantage of this. Now, I'm only giving a highlight into a few of the capabilities. Please note that the tutorials that we're having every single day will expand and do a deeper dive into many of these capabilities. Later today, Trevor will be doing a tutorial on cryo-EM. Tomorrow, Ying is doing a tutorial on single cell proteomics. Wednesday, Moe is giving a talk on tutorial on native mass spectrometry. And on Thursday, Yvette is giving a tutorial on scientific computing capabilities that we have. Now, in addition to this, Steve Reichow in his talk earlier mentioned the Pacific Northwest Cryo-EM Center. This is a joint project between Oregon Health Sciences University and us, PNNL and EMSL, in order to build and operate a national center for cryo-EM. So this is also an available approach for anyone. The center has 5 different microscopes. Basically, the new microscope that we just brought on board here at EMSL for DOE specific work is identical to the top end line of instrument that we have at PNCC. But you can see just an example of some of the structures that have resulted from the use of PNCC in the last year. So this is also a user center that's available to us. And since we jointly operate it, I'm happy to answer any questions as well. Now, how can I leverage multiple capabilities to address a challenging science? This is a question that we wind up getting a lot from users. And so for as an example of what can be done by combining all of these things, if you wind up thinking about this project of PDX 1.2, 1.3, these are 2 proteins involved in vitamin B6 biosynthesis. PDX 1.2 is a pseudoenzyme that's not active. PDX 1.3 is active. And so there's questions of how does PDX 1.2 regulate the activity of PDX 1.3. Their proposed mechanism was a stacked ring of hexamers. We wound up being able to use cryo-EM in order to look at both PDX 1.2 as well as PDX 1.2, 1.3 co-express complexes. Wound up seeing similar shapes, not a lot of differences between them, except for the overall Coulombic charge inversion that you would see. But that really didn't explain everything on its own. So combining the cryo-EM with cell-free expression allowed us to really control the amount of protein that was generated for each one. And you wound up seeing on the left-hand side the SDS page showing both proteins present, but in the co-express complex that they're merging together in various stoichiometries in order to create these stabilized complexes. Now, we were able to do functional assays looking at that to see the effect of PDX 1.2 on the overall activity of PDX 1.3. And this again gave rise to questions. So we wound up referring to native mass spectrometry, utilizing that as a central complex to look at and show that we can get a dodecamer complex out of it that with very high mass resolution. But if you compare PDX 1.2 and 1.3 versus the co-expression, instead of having a single co-complex of six stoichiometry units of each, we wind up seeing distribution of overall co-expressed products. And so this really supported the native page results that we saw early and really redefined the way that you need to think about this model. Instead of these stacked rings, it's really a stoichiometric population difference. And we're looking further into trying to be able to fully characterize these as well as understand the detailed mechanism of the action of assembly for PDX 1.2 and 1.3. So this is just one version of a science example that's taken advantage of all the various capabilities available through EMSL. Now, I've talked about what's currently available and kind of what our vision is. And in the near term, we wind up having two instruments that are coming online this year. The Aquilos 2 on the right-hand side was fully installed at the end of September. We're doing commissioning and calibration experiments as we speak. We expect to have that available to EMSL users already in November timeframe. The one on the left-hand side, the cryo X-ray nanotomography system for whole cell imaging at 30 nanometer resolution, but very large fields of view. This will likely be coming online for users in the May timeframe. Beyond those direct near-term ones, the larger roadmap that we alluded to earlier is to enable visual proteomics. And this is where kind of the overall workflows of everything that I've just described comes into play. We have all these capabilities from native mass spec, NMR, cryo-EM in order to solve various structures of single particles, basically generate a library of template structures that we want. We have the new Krios in order to do ultra high resolution 3D tomography of whole cells. And then using the advanced computing capabilities as well as the single cell proteomics to help quantify overall proteins, and combining the template structures with the whole cell tomography, and then template matching and label-free matching to be able to do really visual proteomics 3D mapping as has been described previously by Beck et al. But doing this now on an expanded scale where we're being able to bring to bear all of the native mass spec capabilities and NMR and single cell proteomics capabilities in order to expand upon what used to be just a cryo-EM focused workflow for visual proteomics. And so with that, hopefully you've gotten a little bit of an introduction to where we're wanting to push the user program in the next 3 to 5 years, and also kind of some additional background as to the overall structure of the meeting this week and why this is an interesting topic for us.